When we're working on and developing bioregenerative life support and we're considering humans in these life support systems, we think about that as we would be thinking about any human on this planet. What do you need in terms of food, water, waste, clothing, toothpaste, medicine? Biosensing specifically is where you integrated a biological molecule or a biological element that's actually going to be the primary detector. Program cuts that happened in 2004 were devastating, absolutely devastating. They completely cut physical sciences and space biology and completely cut even the human research program to zero. I believe that the future of human civilization involves space exploration. And humans are going to move out into our solar system and hopefully beyond at some point. And it's going to require this type of knowledge, this type of mastery of biology.
Welcome to the Omar Alayli podcast, your go-to podcast for exploring the diverse fields driving space exploration and technology. I'm your host, Omar Alayli, and today I'm thrilled to be joined by Marshall Porterfield, a distinguished professor of agricultural and biological engineering at Purdue University. Marshall is also the former NASA Division Director for the Space Life and Physical Sciences. His pioneering work is at the very heart of solving one of space exploration's greatest challenges: how to keep humans alive and thriving on extended long duration missions. Marshall, it's great to have you on the podcast. Thank you for making the time.
Thank you, Omar.
Marshall, your work is at the intersection of biology, engineering, and space science, and you've advocated for an integrated approach you term Biosystemics. This integrated approach is in service of a larger goal: developing a bioregenerative life support system, or BLiSS. In simple terms, this is the concept of creating a self-sustaining ecosystem to produce and recycle food, air, and water for astronauts. A recent paper you co-authored argues that to make these systems reliable, we need to move beyond treating them as black boxes. This implies a need for deep, real-time understanding. So let's start with Biosystemics. For our audience who might think of life support in terms of tanks, filters, and chemicals, could you first unpack what Biosystemics means?
So I started using the term Biosystemics to try to capture really the integrated systems approach that we were really developing as we were also starting to develop omics technologies and omics platforms for doing life sciences research. But really, this is, we're focusing on life sciences and spaceflight, which has some challenges that omics and omics technologies actually offer some advantages for spaceflight. So building a life support system that's made out of living components with a human being inside of it is a very big challenge. And turns out most of the parts in that system are actually all biological. And the biological parts all operate based on the central dogma: DNA, RNA, protein. So we know how biological information flows. And that's really that was one of the really inspirations for Gene Lab, was really the idea of being able to track that biological information flow from information to action, and being able to understand how to connect those actions as biological activity to all those little parts.
Okay, can you explain why is a holistic systems level view of biology an absolute necessity for humanity to survive and thrive on long duration missions in space?
Well, we look at an ecological approach for humans because we know that we're going to have to integrate life support in the form that we use it and that we interact with it here on the planet. We don't actually have to think about the life support system on the planet because it's already operating, because all the plants, fungi, organisms in the ecosystems have grown and evolved over the 4 billion years of the history of this planet to create a balanced, somewhat balanced and stable ecosystem with the right amount of oxygen, the right amount of water, the right amount of CO2. Everything's well balanced for us in this system. And it really is a product of 4 billion years of biological evolution. And because of the complexity of that, we think that an ecological approach is going to be more valid than a physical chemical approach. And there are a lot of reasons that justify that. And first is that biological systems are more or have better performance. They're more capable than most physical chemical systems are at almost everything. Now, there's caveats for that, and there's caveats for how and where and when we're going to use physical and chemical systems in a controlled ecological life support system. But mostly those are going to be used for adjusting for and accounting for the large scale buffering that the terrestrial ecosystem gives us now. So we don't have to worry about the differences in what's going to happen in between fall and winter, summer and winter, in terms of are there going to be enough plants around to make oxygen for us, right? But, well, at the global level, there's enough oxygen, everything's buffered, where those things don't matter so much on a seasonal basis. But we are going to have seasons. We're going to have cycles in these advanced life support systems, and there are going to be needs for buffers between different components in these really complicated systems.
Okay. So if omics give us the biological blueprint, you argue that real-time biosensors are the missing piece, where biosensors act as the nervous system that tells us what's happening moment to moment. How does integrating this live data fundamentally change our ability to manage these complex biological systems, moving them from unpredictable to reliable engineered life support systems?
Yeah, when I started thinking about the ideas that led me to conceive of NASA Gene Lab were actually based on my own work in biosensors, where we were using biological molecules, enzymes, and we could link those with electrochemical systems. So in a sense, we have a hybrid sensor where there's a biological molecule that can transduce a signal to an engineered system that you can read out. And when I started looking at genomics and the emergence of modern gene sequencing, which was actually being reapplied to RNA and sequencing RNA in a way to measure those signals that are flowing from biological systems. I really looked at RNA-seq as really a highly parallel biosensor that's really capable of measuring not just one or two or three of the biological targets that might be in a biological system, but literally measure all of them. And in some ways, you don't have to have a hypothesis anymore if you can measure every factor, right?
Mm-hmm. Yeah.
So by moving to that type of technique, we can really take a whole systems approach, a high content, high throughput approach, where we have a biosensor for every single gene. And then from that, we'll be able to read the input and output of every single gene in that organism or in that cell. And from those patterns of profiles of those gene expression patterns, understand what is the status, disease status, or the basic status of the organism in terms of stress in the environment. And then start to be able to use those readouts from biological systems as a way to readjust and control the different parameters in a bioregenerative life support system. So really, the omics becomes a universal reader. It becomes the ultimate universal biosensor. In some ways, RNA-seq is kind of like a tricorder in that you can really measure every single signal there is, but then discriminating that becomes the next challenge. And that's when we really started thinking about adopting and using AI, machine learning types of technologies.
Okay, and when you refer to biological systems, you're referring to humans only, or is it also plants?
No, when I talk about biological systems, I'm talking about everything that has DNA in it. Plants, animals, microbes, and all organisms share in the same biomolecular platform of information, and they all have the same exact coding system, at least on the planet Earth that we know of.
Okay, so ultimately the goal is for BLiSS to achieve maximum efficiency and resilience. Can you walk us through how this complete data picture from the genetic level to the real-time molecular flux allows you to truly optimize a habitat, not just for survival, but for the peak human performance in the harsh environment of space?
So you can think about this in some ways in components, because we will have to operate some things in components. There'll be a human medical and human performance team that will be involved with working with the human and basically taking a genomics approach, personalized medicine approach, where that person's genome is already sequenced, is part of a data system. And that way, when you take a blood sample or a tissue sample and you read out to see what genes are being expressed or not expressed, that's matched to the individual's individual genome.
Okay.
So that individual also has its own microbiome in our gut and in our skin. And that microbiome is typically would be monitored or measured using hyperspectral sequencing, which is still based on the same RNA-seq techniques,
Mm-hmm.
but you're actually taking a subsample of the human with regard to that. But then we'll also be taking soil samples or samples from the agricultural compartments
Mm-hmm.
or from the bioreactor systems and in the waste recycling systems, and looking at the microbes and the microbial communities in those places and seeing what genes and activities they're expressing. So we'll be able to have basically a complete ecosystems picture of the human and their microbiome as that microbiome interacts with the agricultural system, which would basically be a large-scale farming, probably mostly with hydroponics and microgravity,
Mm-hmm.
but could use some kind of regolith-based systems. But all the fertilizers and everything that are feeding all those plants are derived from the waste stream that came from the human. So there's
Mm-hmm.
going to be a continuum of the microbiome between these different components, just like there is on Earth. Our gut microbiome is essentially, in some ways, continuous with the soil microbiome of the planet.
Mm-hmm.
And then we are also re-inoculated back. Our microbiome is re-inoculated back through our own foods.
Mm-hmm.
So it's not a set. I mean, the microbiome is not set. It's dynamic. It's changing with the human as they age and grow through their lives, but also as they're encountering stress and dealing with different types of environmental stress, which may be triggering pathology and disease. And we'll be able to learn all about that. That's the forefront of medicine today, is
Mm-hmm.
actually understanding genomics and genetic-based information, bioinformatics approaches for precision medicine, personalized medicine. That's really that's at the forefront of medicine today. And in some ways, we were leading in that when we initiated Gene Lab and the twins experiment
Mm-hmm.
a number of years ago now. Yeah.
Cool. Marshall, you've described the need for real time data, as we said, to make these life support systems truly reliable. This brings us to the core of your lab's work, the sensors themselves. While the International Space Station has thousands of sensors for its hardware, yeah, as we discussed just now, you're focused on sensing living systems, biological systems, hence the biosensors. What is fundamentally different and more challenging about building a sensor for a plant or a human compared to one for a fuel tank, air analysis, or a solar panel?
Well, sensing and biosensing, biosensing specifically is where you integrated a biological molecule or a biological element that's actually going to be the primary detector. So
Mhm.
enzymes make very good biosensors because they have such very high specificity for the target molecule that they operate on. In some ways, enzymes have some of the best, molecular specificity that you can basically that you could hope for. And then if you can couple that enzyme to the output of an electrochemical transducer, then you can have real-time measurement of what that enzyme or biological system is doing. So, depending on the application or where you're going to use the sensor, whether it's in the body or in a wastewater treatment stream, you have to package it so that you're basically protecting the biological component, the enzyme, so that it doesn't become degraded or broken down. So in terms of genomic sensing, that's different because really we're relying on sequencing enzymes that we can use that will actually identify the DNA and RNA and then make copies of it. So we actually have in that kind of biosensor scheme, the enzymes, the transcriptases are actually the biosensors that are recognizing the biological molecule that needs to be measured. But with those types of methods, you have additional steps that prevent you from having true real time. But those are some of the technology areas that we're interested in developing now, is increasing the throughput and the response time of RNA-seq type of sensors, so you could have more real-time biological response measurements, looking at the genetic responses.
Okay, can you expand on what RNA-seq is?
RNA-seq is a type of nucleotide sequencing technique, which is really a derivative of the original DNA sequencing, but it allows you to sequence RNA molecules to make copies, and that RNA-seq and when it makes a copy, it's making copies that will be detectable by an analytical reader later on. So it's a type of analytical technique that allows us to measure and quantify the amount of gene expression that's happening in a sample, and it can discriminate from every single different gene.
Okay, so your lab has produced some truly futuristic technologies like the nanotetherball biosensors. If we think of a biosensor as the nervous system for a habitat or a spacecraft, we could say that you use biomimetic as the hardware and the nanotechnology as the advanced material. First, could you define what biomimetic and nanotechnology materials are from your perspective? Then explain how these pieces fit together to help achieve an all-purpose sensor platform?
Yeah, so biomimetic refers to a collection of technology that are based on microelectromechanical systems. So that's an acronym that's used in the silicon microfabrication world. That term was first coined to talk about microscale devices that you can make out of silica or with etched metal. Basically derivatives of a lot of microfabrication techniques that are used for making microchips. A biomimetic is a MEMS device that's been specifically developed for biological application. And in these, we typically incorporate fluidics and small chambers for cell culture that can be integrated with specific types of sensors in those chambers. So these can be set up where you could put a MEMS device literally as a lab on a chip, where you could take a blood sample and it's possible to take that, expose it to the chip, and the chip would actually be able to take in the cells, potentially sort different cell types, and pick the cell type that you want to measure and actually do a measurement on it and give you the output from that. Usually either through optical or electrical measurements that can then be transduced by the device.
And what about the nanotechnology materials that are being used?
So yeah, and then these MEMS devices typically have features or elements that are on the micron scale or submicron scale, and a lot of the technology we've been working on does push beyond the micron scale into what is referred to a lot of times as nanotechnology. So we've been working on different devices where we've been integrate DNA and other types of components, and really when you're integrating at that molecular scale, that's really at the scale where where we usually use the term nano to describe that it's really, really super small at that point.
Okay.
Yeah.
So you've talked about the biomems and about the nanotechnology. So can you put them together? What about the nanotetherball biosensor? Do you have? Can you speak a little bit about it?
Yeah. So when we're actually creating some of the more exotic materials that we actually make a lot of those using electrochemistry and combinations of electrochemistry with other types of material techniques, but especially with electrochemistry, we can create and write fairly small features. And what we've been able to do with that was we would take a MEMS device, like a silicon chip, and have an area on the chip that's functionalized where we could grow carbon nanotubes. So the chip itself is just like just a platform that we can plug into. And then plugging into that gives us access to this little micro region on there where we can grow these nanomaterials. So we actually grew the nanotubes up off the surface of the chip. And because the chip had an electrical connection we could connect to, we used those nanotubes to electroplate these, what ended up being nanocubes, palladium nanocubes on the carbon nanotubes. So, you're talking about definitely a sub nanoscale or near, super nanoscale types of devices. And with those little cubes, we're able to actually functionalize those with enzymes.
Okay.
So we have this little carbon nanotube that's, just, a few carbon molecules wide and around. And on it is a nanocube that's, sub nanometer size that we've decorated with enzymes. And then those enzymes, we can actually read the output from the enzymes on the nanocube through the electrical connection through the carbon fiber. Because the carbon fiber is actually electrically connected.
Yeah. Okay. So what about the self-referencing microsensor technique you're developing in your lab? What is it and how does it allow you to see that which was once previously invisible in real time?
Yeah, so there's different approaches we use to build sensors and use electrochemistry to study biological systems, especially at the single cell level. We're trying to, one approach is just to get as small as you can. Another approach is not just in having small sensors. Turns out it's how you operate the sensors that becomes just as important or more important than having a small sensor. Because when we get sensors down to a micron or size and a tip size, like these micropipettes we make that are basically little microneedles that we can take down to a single cell, and we want to measure what the cell's doing. And since we really want to measure the transport at the cellular level, so we use a technique called self-referencing, where the probe is actually oscillating at a fairly slow rate, about 0.3 hertz, that we don't want to mix, but it can also actually oscillate back and forth near the cell, so we can sample far, near. And if the cell is releasing something out, there should be a concentration gradient between the near pole and the far pole. And even at the slow frequency that we're moving this probe, the fact that we're inducing a waveform back and forth, we're producing an oscillation into the signal, then all of our filtration happens at that frequency. And it turns out that even those slow frequencies can filter out a tremendous amount of noise. We can get, nanomolar to picomolar level flux measurements at the single cell level. And those signals of the flux signals that we can on the waveform on top of the probe are way smaller than what the noise would be on the sensor. So essentially we can use that as a trick to measure flux and improve our resolution at the same time. And it turns out measuring flux is much more important than just having a concentration measurement, because flux is actually active transport. We're measuring the active transport of the molecules that the biological system is creating. And measuring flux at those levels with that level of filtration allows us to really see aspects of cellular physiology that normally wouldn't be possible.
And why is it important?
Well, I can tell you, I can give you some examples of how it has been important. For example, with that technique, I was able to do some measurements with macrophage cells, which are cells within our immune system that make, when they become activated, they'll produce peroxide and nitric oxide, and they attack and try to kill viruses. Well, I was able to go down at the single cell level and measure sustained nitric oxide flux from macrophage cells. And also, too, we've been able to do recordings of oxygen flux associated with neurons, and also with beta cells, which are involved with secreting insulin. And the experiments that I did with the beta cells and it's secreting insulin, when we triggered the cells to secrete insulin by giving them glucose, and with the probe we're measuring the oxygen flux from of the mitochondrial activity of the beta cells as they're starting to secrete insulin. And then what we were able to see in those in the oxygen flux from the beta cells is that it actually oscillated. So there was a periodic activity of those oscillations that was correlating with the release of the insulin vesicles by the beta cells. So we're able to correlate the temporal properties of insulin secretion to mitochondrial oxygen activity by measuring oxygen flux at the single cell level in these systems. And that knowledge allowed us really to understand that the regulator in terms of clutching those secretion events actually is the mitochondria in the beta cells.
Okay, fascinating. So let me ask you this, let me follow up. What has been the most surprising discovery you've made by being able to measure life at this dynamic molecular level?
We've done a lot of things, it's we've done a lot of work in cancer, in breast cancer, and really verified and validated a lot of what was starting to emerge in terms of the metabolic models of cancer, in terms of mitochondrial dysfunction. So we actually did a lot of work combining the oxygen, our self-referencing oxygen sensors with the self-referencing glucose biosensors, being able to measure oxygen and glucose and the Warburg effect in cancer. That was, we really verified that in a number of systems. Also, we did some of the first recordings and measurements of nitric oxide flux associated with pollen tube growth and plant reproduction. We actually did some experiments at that time, were some of the first experiments with plants where we used Viagra to show we can modulate the nitric oxide signaling in the pollen tube. So that was really, really interesting at the time. We could take these pollen tubes that'd be growing normally, and then you put Viagra on them, and they stop growing. They just swell up until they pop.
Okay, very interesting.
So there's a similar effect in plant pollen tubes with Viagra, we discovered. I don't know how big of a discovery that was, but it's definitely interesting.
It is very interesting. Marshall, your team has developed a range of novel sensors from modified nanocomposites, like the graphene-based ones that can detect glucose in saliva, tears, and urine. This sounds incredibly complex. Could you take us inside the innovation process? What does it take to go from a theoretical concept to a functioning nanoscale device that can reliably detect a single type of molecule in a complex fluid like blood or water?
Yeah, so we've basically been able to create a platform for biosensing. We have several different types of materials that we can use. One that I really like to use is one called platinum black. It's an electrochemical nanomaterial we can grow, and normally if you electroplate platinum nicely, it becomes shiny and smooth. But I can electroplate it in a way where it becomes all hyper the surface area is hyper activated, and it appears black. And in all that electrochemical surface area, we can basically get it to hold on to enzymes. So enzymes like glucose oxidase and glutamate oxidase. So we can measure neurotransmitters, ethanol, lactate. There's large numbers of biological molecules we can sense with these different types of enzyme-based biosensors. And designing those are really a matter of trying to just determine whether or not the conditions that we can immobilize the enzymes are going to be conducive with the electrochemistry, designing the electrochemistry, because there's different types of electrochemical reporters we can use to couple to different types of enzymes for biosensing. But really the main driver for me and when a lot of those applications is really the science, because I you know, my pathway is different because I basically have do hypothesis-driven engineering. So if there's a critical problem in biology or in medical sciences that there's a limitation in terms of the experimental measurements that you need to make, that's really what I get. That's really what I like to focus on. I like to focus on the impossible experiment that can be enabled by a new technology, and then go after developing that technology to enable the science. If you look at the history of science, it's really that's the unwritten story behind all of the discoveries in science. Because not a few hundred years ago, people were working with lenses, and they were looking at the heavens and trying to observe the stars. And then one day someone figured out if you grind those lenses and turn them down into a drop of pond water, what would you find in there? Well, they found a whole new universe. They found a whole new field of biology called microbiology was born from that new tool. And it's similar. If you look at across biology, and when there's big surges of experimental growth and advances, those are usually associated with new technologies. The electron microscopy also did that, but really radiation, the discovery of radioisotopes and the ability to work with radioisotopes unlocked all the fundamentals of basic metabolism and biochemistry, and leading into the ability to track and monitor DNA molecules. So all those technologies add up, and they all have impact in terms of the advancement of science.
Speaking of innovation, biology and engineering, one of your innovations is a biomimetic root oxygen sensor. This suggests you are learning from and mimicking biology itself. How does nature inspire your engineering, and why is a biomimetic approach often more effective than a purely mechanical one?
So the biomimetic approach is interesting because with that sensor system, we're actually measuring oxygen flux. And the way it's designed is that we have a sensor that's essentially, if you took a real root and you made an exact copy of it, except for it wasn't a real root anymore, but it had the same size and shape, but it would consume oxygen just like a real root would. So in this little cylinder, it's consuming oxygen just as a real root. A real root, if it's in the environment, whether it's flooded or there's microbes that are growing around it that are consuming oxygen, those are things that can impact the oxygen bioavailability to that real living root in that system. So what we figured out is that if we recreate the geometry and shape and profile of a real root, but with the same level of oxygen consumption, we could actually measure the oxygen consumption biomimetically that would tell us what a real root would sense in that system. In microgravity, where buoyancy is inhibited, there's no airflow like a candle flame. The candle flame will go out. It doesn't burn as a flame, it burns as a little sphere. And because it's burning as a sphere, it'll go out in space. And that's because it's completely diffusion limited. Without buoyancy causing hot air to rise and bringing in cool, fresh air, without all this airflow, the flame won't look like a flame. It looks like a sphere. It turns, because it's diffusing from all around. The oxygen's coming from all around really slowly until it goes out. And that's what the problem we needed to solve in space. There's no difference in oxygen concentration in space, but there's a difference in oxygen bioavailability because there's no buoyancy-driven mass flow in space. A candle flame will go out. The plant root will become hypoxic. And that's what we discovered in my original work during my dissertation. I was analyzing plant roots from the Chromex experiments, and we were finding that they were responding in space just as if they were being flooded.
Okay.
Right?
Yeah.
Well, they're not being flooded. Why is that happening in space? How do you prove this biophysical hypothesis? That's why we actually built the biomimetic ROB sensor, so because it would exactly mimic a real root. And so we actually flew the ROB sensor in the Vomit Comet, the KC-135, the plane that flies the parabolas.
Mm-hmm.
And when it flies at the top of the parabolas, when it kind of goes over the hill, you get microgravity for a little bit, for about 20 seconds
Mm-hmm.
when you're falling. In that 20 seconds, what we saw was the oxygen bioavailability to the ROB sensor would go shooting down. And then as soon as the gravity would turn on and go back to 2G, it just immediately shoots back up.
Mm-hmm.
So the sensor's responding in real time to the bioavailability of oxygen in the environment and the concentration, but the bioavailability is changing because of the diffusion environment in microgravity. And that we were able to directly measure that with the ROB sensor. Yeah.
Very interesting. Okay, so for a biosensor to be useful in space, it has to meet incredibly high standards for sensitivity, selectivity, and biocompatibility, all while being miniaturized. From an engineering perspective, which of these parameters presents the biggest challenge when designing for a multi-year Mars mission, and why?
In spaceflight conditions, most of the time there's not going to be a researcher who's going to be able to really work with that sensor in a way that you would in a lab. So that's one of the main constraints, is that the sensors almost have to be foolproof to some extent, or they have to be built into a system where you've got adequate controls and fluidics, where you can really have those kinds of parameters covered. So really, those are some of the main issues, I think, with working with some of these sensors in space flight. They are going to be in a lot of different potentially be in altered atmospheres in terms of the atmospheric pressure, and issues like that can change over time. But they really need to be robust. Calibration. One of the biggest problems with any type of sensor is calibration drift. Over time, their calibrations will change. And how do you get the sensors back and recalibrate them at the end of the experiment? Those are the issues. Those are the issues that limit those kinds of experimental sensors. But there are a lot of different types of physical chemical sensors that are used on station fairly robustly over time, that are fairly stable. But when you start looking at more specialized biosensors for research and these types of research applications, it can become very challenging, especially when you start thinking about the biological components of a biosensor, because those elements can actually potentially be affected by the radiation environment too. So that is that introduces a new factor in space that we normally don't have to contend with, is what is the radiation effects on the sensors. And that will potentially become a much bigger issue as we go out beyond LEO and we start to do our next phases of research and exploration on the moon.
Okay, so let me quickly follow up on this. How do you test or plan to test a device like that to ensure it will survive a long duration journey and survive, like you said, extreme space environments and work reliably millions of miles from home?
Well, there's standard testing protocols for any type of anything that flies in space in terms of its radiation tolerance, pressure, temperature, all those sorts of issues. Everything also goes through a shake test too. So they have these giant shake tables. You can put whole systems on and shake them and make sure they're not going to fall apart during the launch. So a lot of this is the mechanical stability.
So do they fall under the technical readiness level testing, like phases, similarly, or
Everything does. Everything you fly up into space will have to, will fall
but
under that.
even that stuff that have biological components?
The biological components would probably be, unless they are potential contaminants.
No, I'm talking something like enzymes, like
No
or is this
enzyme
something
the device. It would be it would have to be the complete device.
Okay.
If you had a sensor that could be configured with multiple different enzymes, probably the sensor would be certified, and then you could configure it with the different enzymes. You wouldn't have to recertify it with every enzyme.
Okay, cool.
But they
So,
are very strict about what can fly
mhm.
and TRL levels, and if you have any kind of, if it's flown before, if it has flight legacy, that's usually you're A plus. You're good to go.
Cool. So, looking at the challenge of biocompatibility, when you're designing a sensor for in vivo use inside the living organism, how do you ensure the body doesn't reject it and that the sensor itself doesn't alter the very biological process it's trying to measure?
Yeah, I haven't done a lot of in vivo recordings, but I worked with some people that's some of their main concern, especially in the brain interface area, where they're actually talking about embedding chips in neurological tissue. Immunological rejection has been one of the primary uh, issues with those types of devices. I'm not sure where they're at. What are the standards now in terms of those types of devices and those types of standards now. But that is a major area of concern. Anything you can do to coat the device in any kind of material that would change its immunological signature is going to be important. But that's very difficult because, even every single cell has its own marker that tells the body that it's self. And anything that's not self tends to become compartmentalized. And that is one of the major problems with implantable devices. But, I do, most of my work is actually, experimental, dish-based, or, organismal based types of experiments. So I don't do a lot of that implanted type of work, but the rejection issue is the major concern there.
Cool, fair enough.
But we can, but if you can do non-invasive measurements using self-referencing, and you're actually measuring flux, the advantage of not putting the electrode or the sensor in the organism is so much above what you can get with implantable devices. That's why I've never really focused that much on that. I'm focusing more on making the device stable and compatible for long-term use, not necessarily in the body, but for more non-invasive types of applications.
Yeah. Marshall, like you're saying, yes, your work is pushing towards non-invasive health monitoring, like detecting disease markers in saliva, tears, and urine. Looking beyond glucose and oxygen, what is the next major biomarker you believe we need to be able to track non-invasively? And what breakthrough in sensor technology is required to make a mission to Mars a reality for astronauts?
Well, it would depend on what your application is.
Okay.
Probably for human medical, in blood, glucose, lactate is usually a good indicator of there being anaerobic or hypoxia in the system. So probably lactate. We've done a lot of work with glutamate in the past. There's a lot of really dynamic and important applications of that in neurophysiology, and also looking at combining that with oxygen sensing for Parkinson's and other types of neurodegenerative diseases. It's really multi-sensing, I think, is more important now, because if you look at one element, usually there's another element you want to look at with that. In neurophysiological systems, you can actually measure a lot of neurotransmitters like acetylcholine directly with electrochemistry. So you could do experiments where you're looking at glutamate and acetylcholine with oxygen, combining ions. pH is really important because of the role that pH plays in redox status of cells and living systems. And in terms of primary metabolism and primary physiology and health, I still think measuring oxygen and pH fluxes are the primary starting point, especially from a biophysical perspective, because you're going to have massive amount of redox fluxes associated with primary metabolism, usually oxygen, and there's a lot of proton fluxes associated with that too.
Okay, what breakthrough in non-invasive sensor technology is required to make a mission to Mars a reality?
I can answer that. I can answer the sensor question by saying probably a radiation new types of radiation sensors that could potentially detect some early primary product of radiation stress.
Okay.
And and that might be a question mark because maybe no one knows exactly what that is or what that could be. So I would say, on a mission to Mars, that radiation is going to be one of the primary stresses that we're going to face. And really that's anything that we do from here on out in space. We're going to have to go beyond low Earth orbit, where we've been hiding inside of the ISS, which is also hiding within the radiation shielding of the planet. So crew members that go to space now, they don't experience the radiation that the same crews that accomplished the Apollo missions did 50 years ago. So the radiation is the game stopper in terms of long-term extended human missions to the moon and Mars. Most of the Apollo missions never lasted more than 7 or 8 days. I think at the most 8 days. And those crew members in that time area, of course, it was a different time. But if we look at the health outcomes of the 19 Apollo astronauts who actually left low Earth orbit compared to ISS astronauts who spend 6 months on the ISS, the Apollo crew have 500 to 600% higher levels of cardiovascular disease for 6 days of space flight compared to 6 months on the ISS. So radiation environment is really, really a serious new consideration in deep space. So we're going to need better sensing and biosensing related to radiation and radiation stress responses in human beings. And we just don't know what those are.
Okay.
Because we just haven't had that type of research experience. We haven't had that type of human exposures. Most of our radiation research is done through short-term bursts, exposures. We do radiation like at Brookhaven Labs. You can take cells in, they'll burst them with radiation, and then you go off and see what happens three weeks later. That's not chronic radiation. It's not the same as what we get in space. And for the most part, we don't even know what the full spectrum of radiation is in space because it's a combination of solar and background cosmic galactic radiation, which is really serious in its own right. So I would say some type of specific type of radiation damaged metabolite type of sensor, and then maybe some more better primary radiation sensors that can discriminate maybe between beta radiation versus galactic cosmic radiation, because they have the different radiation species have different biological ramifications. Okay.
Marshall, let's now shift gears to the bioregenerative life support system and explore this area a little bit more. A recent policy paper you co-authored presented a stark history. It states that in 2004, NASA canceled its advanced bioregenerative programs like the Controlled Ecological Life Support System and the BioPlex, and even physically demolished the facilities. In the 2 decades since, China has embraced and advanced these very same concepts, building their Beijing lunar palace. From your perspective, what was the strategic consequence of NASA's 2004 decision, and how has it shaped the landscape of space exploration we see today?
So the program cuts that happened in 2004 were devastating, absolutely devastating. They completely cut physical sciences and space biology, and completely cut even the human research program to zero. And then they later returned the human research program back to about 10% of what its previous funding was, and reestablished space biology and physical sciences in name only. Those cuts were done because of at the national level, they were trying to fund. The U.S. was initiating and starting to fund the next big large rocket project, which at the time was Constellation, but essentially became SLS. So, and during those days when when we complained about it, the argument was, well, you're not going to have a science program if you don't have anything to fly on. So you're just going to have to wait until we, our reinvestments have produced the next flight system that we'll need you then. Then we'll need space biology. But it was really short-sighted because, the requirements for bioregenerative life support and those kinds of capabilities are going to be needed for lunar outposts. We're just not going to be able to, have a resupply rockets flying back and forth all the time. It's just not economically feasible at any level, even with reusable rockets. And the Chinese understood that from the very beginning. And at the time that the U.S. cut all of our programs, that was right when really the Chinese program was just starting. They literally had the first Chinese astronaut flew in space in 2003. In 2004, we were cut. By 2016, the Chinese program had produced the Beijing Lunar Palace and had completed a one-year mission with a crew of 4, with a lot of help from the research and literature that had been produced by NASA in the previous decades. So that's a major accomplishment. That is a world's first, and NASA we have not matched it, and we have not reinvested back in the programs that will allow us to actually do that type of work again. Mm-hmm. We're doing some habitation work now, but it's really the habitats. They're building small, analog habitats, putting people inside and doing basically psychology testing on them, not closed bioregenerative life support. And that's what the Chinese did. They produced all the oxygen, food, and water that their crews needed for a year in a closed system, and that's now a platform that they can exploit with their International Lunar Research Station, and that is considered to be the centerpiece of what they're doing. And if you look at all the research they're also doing on Taigon Station, they've moved ahead in both in the quantity and quality of a lot of the life sciences research that they're doing. Has really been phenomenal. So,
yeah. Okay, so that decision to cancel the controlled ecological life support system and the bioplex has led to what your paper calls urgent critical gaps in the US capabilities. Let's be specific for our audience. Let's say if the US were to establish a lunar base tomorrow alongside the Chinese base, what are the 1 or 2 critical life support functions that the Chinese taikonauts could perform using their BLiSS that the American astronauts rely on resupply emissions simply could not?
Well, based on what they accomplished with their facility, they ought to be able to resupply the majority of food and water to the crew, and also regenerate the most of the atmosphere. Atmospheric regeneration and maintenance of an atmosphere in a station like that is really going to be costly in its own right too, because every sealed system will have a leak rate. Even the ISS has a leak rate, and they have to fly up on a regular basis nitrogen gas just to fill the atmosphere, because 70% of our breathable atmosphere on the Earth is nitrogen gas. They actually have to fly up nitrogen gas to fill the atmosphere to have a 1 atmosphere pressure on the ISS. Maintenance and controlling an atmosphere, the plants are going to be able to accomplish a lot of that, and especially in producing oxygen and recycling and producing potable water too. So there's a lot of advantages for having plants and plant-based agriculture. And you start looking at those basic supplies, even just water. what it would take to resupply water. If you can do that with a regenerative system, that's a huge amount of upmass that you save.
Mm-hmm. True.
And
So,
even
yeah.
that one thing
Mm-hmm.
could mean the difference in terms of the logistics costs for maintenance of a facility like that. Ultimately, it's going to come down to, all right, can you pay for it?
Okay. So your paper notes that the International Space Station has been severely underutilized for advancing this science. Why has this been the case, and what are some of your concerns of decommissioning the International Space Station?
Well, the U.S. program in 2010, the Congressional Authorization Act that year designated one half of the ISS, one half of NASA's resources on the ISS to be a national lab. And that specified that as a national lab, that they would not ever do any NASA related research. So they're doing research for like Brookhaven or Los Alamos National Labs. These are national labs that are run for the government by NGOs,
Mm-hmm.
and they produce large bodies of primary and applied research for our nation and for our nation's agencies, including our defense agencies. And in specifying the ISS as a national lab, gave it that status, which what ultimately did was, in giving all those capabilities to all the other aspects of the federal government, and it excluded it from NASA, and that became actually a huge rate limiting factor
Mm-hmm.
for the amount of research that we could fly. We had one half of the rodent research capabilities that NASA could use, and the other half went to commercial pharmaceutical companies or NIH, or, with the commercial pharmaceutical companies, they flew their proprietary information. So there were, in a lot of cases, you're flying and implementing science that's not adding to or complementing or contributing to the overall mission of NASA. So we lost really half of our entire research portfolio to that. And if you look at the reviews and of what the National Lab has been able to accomplish, the International Space Station National Lab, also referred to as CASIS, the
Mm-hmm.
Center for Advancement of Science and Space, the General Accounting Office, and other federal oversight agencies have reviewed their productivity. In every case, they've been slammed for failing to accomplish their mission. So there hasn't been a lot of transformative or valuable research coming out of the ISS National Lab because of the way it's been managed.
So, will a U.S. commercial space station, as an alternative to the International Space Station, be able to close some of these gaps that occurred over the last 20 years?
Well, we need applied research that's applied towards the development of advanced life support. A lot of that advanced life support work is going to have to be done in a reduced or altered gravity environment. So even Martian gravity is reduced enough that it really does change the way that a flower pot would work in space, right? How the water drains out of it. You have to re-engineer the soil to match the gravity in a Martian environment. So there's just a tremendous amount of work that needs to be done in that environment in order for us to be able to move on. And we just haven't had the throughput in terms of the access to the ISS, but also, too, after the budget cuts in 2004, when Congress has been, in subsequent years, the budgets for the work at NASA in control of environment agriculture or in bioregenerative life support have not been funded. So that funding has not been turned back on. There's still a small, only a, 1/10 or less of what the previous funding was like, and then only 1/2 of that, the access to the ISS. It's been pretty bleak over the last 15 or 20 years in terms of the amount of research and the quality of the research that we've been getting out of NASA and the ISS. And we really haven't been, ticking the boxes for bioregenerative life support because it hasn't even been a priority for the program. Even up until, I would say 3 years ago, people weren't even talking about moon to Mars. They were just doing research just to, for the sake of doing research in space, for advancing fundamental science. It wasn't being applied towards, the problems that we need to understand and solve in order to build reliable ecological life support systems and stick people inside of them and keep them alive.
Okay. So let's get back to a little bit back to the science. To bridge the gaps, we need to understand, like you said, the fundamental threats of space. Your work in mitochondrial biophysics points to a mitochondrial dysfunction as a key issue, mirroring the aging process here on Earth. How does the deep space environment wage war on our cells at this microscopic level?
Well, we've learned over the years it's actually two things. One is the physical environment, the biophysical environment. And I refer to this as indirect effects of microgravity on biological systems, because you can think of the direct effects or the effects that it has on your body, right? You feel lighter, your muscles are unloaded, blood will move up to your head, right? Your distribution of fluids in your body redistributes. And all of those effects of muscle unloading, bone unloading, all that, muscle weakening, bone loss, all those are primary effects. But there's something else that happens in space that actually is in some ways more insidious, and that's we refer to as the indirect effects. And that's the changes not in the matter of our body, it's actually the changes in the matter that we live in. What happens to the matrix that we're embedded in space? And that's what I was talking about before when I was talking about the candle flame. You can think about it in terms of a fish being in water. If the water stops flowing, the fish will probably die. If there's not enough oxygen in the water and the water doesn't flow, that's much more obvious. But actually, we live in a fluid too, right? The gaseous environment is just a fluid. And what we found is that inhibiting buoyancy-driven convection, and when things are diffusion limited, that actually that affects everything in terms of biological transport, because every cell, every organism is trying to exchange redox exchange, gas exchange, nutrient uptake around roots. All those things can be inhibited because there's not any background flow in the system, and everything becomes diffusion limited, which is completely unbiological in terms of the terrestrial world. There's always a gravity, there's always a gradient, and there's always buoyancy and flow happening around you. Even if you don't realize, that light over here is creating all this flow in this room. That would be happening whether I was here or not. So for a human breathing in an environment, if you don't have gas flow, you can actually be entrapped inside of a redox bubble. And that's what our research has shown. We've actually developed a computational fluid dynamics model of the atmosphere and a human being in the atmosphere in such a way that we could turn on and turn off gravity and just see what happens to respiration. What happens to your breath if you turn off gravity? Well, it's actually the same thing that happens if you turn up the heat, because the buoyancy-driven flow around our body is based on our body being hot at 37 degrees Celsius, and the air being something like 20 or 16 degrees. It's really that temperature difference between my body and the air, just like the temperature difference between the candle flame and the air drives buoyancy. And so without buoyancy-driven convection, it's the same as me being in a 98-degree room. My breath doesn't float up. It sticks here, and it just stays in front of me, and I start rebreathing it. So what do you feel when you walk into a warm room? You feel stuffy.
Mm-hmm.
That's why you feel
Fatigue,
stuffy,
and yeah,
because you're
and your
actually
performance.
rebreathing your own air.
Yeah, creating an invisible carbon dioxide bubble.
Yeah, you end up you're trapped in a hypercapnic bubble. And really, the reason we didn't really understand this until now is because there was no fundamental model of the human body. And that really what it came down to. In order to solve the problem, to understand spaceflight hypercapnia, we had to actually just build a fundamental model of the human body at 37 degrees in a room at 22 and see how the airflow goes around it. Well, I can show you some of those models if you're interested in some of that. I can show you some of it later. We actually have a paper in review on that now.
Perfect. Yeah, I'd love to read it. Can you give an anecdote? Has this ever happened with an astronaut where they caught in this bubble, and what symptoms did they show?
Oh, every single crew member who's been in space has suffered from this in one way or another.
Mm-hmm.
There's different levels of awareness, I'd say, that's developed over the years because it's never been officially acknowledged.
Mm-hmm.
Although I know that there are some folks in the flight surgeon's office who think this is one of the most important problems in space, and that being trapped in a hypercapnic environment and that persistent CO2 rebreathing does create a lot of long-term health problems. Immediately you have a headache, but even if you're in, background on the Earth is the air is about 400 parts per million. If you're in a room where it gets up to 1,000 parts per million, even then you can start to have cognitive impairment.
Okay.
1,000 parts per million CO2 will cause, cognitive impairment and decreased mental performance. So, so on the ISS, even with the scrubbers can bring the CO2 down to about 2,000.
Mm-hmm.
So their starting point is already super high. And then if they're rebreathing their air, they're probably rebreathing at 4,000 to 5,000 half of the time. If I talk to crew members who are aware of this and they're familiar with it, they tell me that when they're working on the ISS, they always make sure that their face and head is near one of the vents, so they're getting airflow on their face all the time. But this is really a major issue in space flight, and when you could read Scott Kelly's book too. He talked about the CO2 on the ISS, and he was very sensitive to it, and he could sense it as it would start to get, as a scrubber would need to be replaced, and he was usually the one that was replacing the scrubber because every time it got high, he would get headaches, and I think he called CO2 the bane of his existence on the ISS. But really, it's I believe it's an unrecognized environmental health factor in our human-built environment. I believe that if you look at, and I've done a lot of different things. I have CO2 sensors I wear, and I have them up in my labs. And if you hop in a car with your family, start riding down the road, you'll be at 3,000 or 4,000 parts per million within 15 minutes if you don't roll down the window or turn on some kind of ventilation. Why do people fall asleep and drive off the road and kill themselves every year?
Okay.
CO2.
Wow. Yeah.
Why is, if you go to an expensive apartment building and you go up to the top floor, they almost never have to run the heat up there because all the hot air rises.
It rises up, yeah.
Guess what else rises up there? The CO2.
Mm-hmm.
They're living in a CO2 nest on the top of these buildings. CO2, long-term exposure to CO2, I believe, is responsible for cancer, neurodegenerative diseases, and it also exacerbates metabolic syndrome and diabetes and those kinds of conditions. So space flight, there's two factors. One is the microgravity factor, and we're back breathing our own CO2, and that's causing mitochondrial impairment directly that way. But the other way is through the radiation itself. Now, we're lucky on low Earth orbit that there's not high levels of radiation. But there is enough radiation up there that it does cause radiation damage and radiation responses that the body can mostly overcome. So it's enough radiation that hurts you, but it doesn't kill you. But while it's hurting you, what it's doing is it's causing mitochondrial impairment.
Yeah, damps.
So this is what we believe is happening with the physics of microgravity, is that the radiation is interacting with these energetic membranes. Mitochondria have all this electron transport and all these biophysical processes that are really important for making ATP. And then in the mitochondria, oxygen acts as the electron acceptor. So we burn our food, we strip the electrons away from our glucose and our candy bars, and the electrons go into oxygen to form water. And along that way, if they don't flow properly, they'll form reactive oxygen species that can, in the long term, cause disease and impairment.
Okay.
And it's really that double whammy of the hypoxic microgravity environment because of the microgravity diffusion stress and the radiation stress, and both of them are going through the mitochondria. And so it's potentially a double whammy in terms of, so if for crew members who go up and fly on the ISS, how does the atmosphere and how does all this affect them? Well, when the crew member, before the crew go to space, they do all kinds of practice of all the experiments, all the procedures, and everything that they could do that they're going to do in space, and it's all timed and controlled and monitored. But when they go to space and they have the same task that they do on the ground, they always give them twice as much time to do it.
Wow.
Because they have the space stupids, and that's the unofficial name that they call it.
Didn't know that.
So every crew member who goes up is impaired by the microgravity environment, partially by the atmosphere, partially by radiation. To what extent, we really don't know yet. But I think we're getting closer to understanding some of these major issues now. I really do.
Yeah, me too. So let's shift gears now to fungi. Your research also extends to a less discussed kingdom of life, the fungi, as I said. You're studying the fungal gravity responses, how fungi respond to gravity, and you're also developing mycoponics to grow fungi in space. Beyond being a food source, you've patented methods for producing pharmaceuticals from fungi in space at a technological readiness level of 4. What fungal gravity responses are, and how do fungal growth behave in microgravity of space that makes them special?
Yeah, so the fungi, most of their gravity responses are really limited to the fruiting body production. So we're not really that concerned with the gravity responses of the fungi, just in terms of having to grow them. And actually, to some extent, we don't really have to worry that much about plants, because if you have water in the right place and light in the right place, they respond to those gradients. And gravity is not really a showstopper for plants either. And turns out we're more interested in the biophysical environment and trying to facilitate redox gas exchange and water uptake and nutrient uptake. But especially, for gas exchange in plants, in the microgravity environment, where in root systems, we've seen hypoxia responses because of the microgravity environment, because they're not getting enough oxygen. And now there's new data that we're actually getting ready to publish a paper that describes photorespiration in the shoots, which is a similar type of biophysical limitation and gas exchange happening in microgravity. Now, with the fungi, they're going to respire. They're able to take inedible crop residues or other waste materials and metabolize that, but they're highly aerobic organisms when they do that, and they're going to need to have oxygen. So essentially, when I started thinking about what it was going to take to grow mushrooms in space, I started looking at what we did with plants. And one of the most successful approaches for growing plants in space with hydroponics is the porous tube plant nutrient delivery system.
Mm-hmm.
And it's essentially, I did part of my dissertation work. I worked on the porous tube system as part of my work at looking at plant root responses in microgravity.
Mm-hmm.
So what I was able to do is just take that platform and adapt it so that we could actually grow fungi with it.
Mm-hmm.
And so basically what it is, it's a porous ceramic tube that has size exclusion filtration built into it, and other materials that allow us to basically feed what would normally be contaminated media into the tube, and then it's actually decontaminated by the size exclusion and the filter properties of the materials that we have in the filters, so we can deliver clean media out to the fungi that grow on the tube surface. And I can show you pictures that show the tube that's completely, covered by the mycelium, and then how that mycelium actually can form primordia that'll form mushrooms and grow off of it. So the mycelium is really just a collection of filaments, and the filaments are actually the proper body of the mushroom. Is the dikaryotic filaments. It's all these white threads you see growing. That's actually the mushroom body. And it's not until they actually grow the fruiting body to reproduce that you actually see a more complicated form. So really, in growing mushrooms, really we're growing mycelium. And the mycelium is just like a mesh network that covers over the tubes and grows over them, and then forms mushrooms. But, we can grow mushrooms with the mycelium. In some cases, the mycelium is a source of a lot of drugs. So the mycelium, we've been able to observe the mycelium producing these exudates, which are sources of new types of compounds that actually are not available with normal other types of methods for growing mushrooms. So we have this whole exudate system we're starting to explore as new sources of antibiotics. But there are large numbers of pharmaceuticals that are produced from the mycelium of fungi. So about 18% of the world's pharmaceuticals, like cyclosporin B, is and a lot of cardiovascular drugs, like statins, are directly produced only in fungi. So about 18% of the world's pharmaceuticals today are produced from fungal mycelia. So what they do is they grow the mycelia either in liquid culture, and then they just harvest the mycelium and extract the drugs, or they grow it in some type of solid culture, and then again they have to harvest the mycelium and extract the compounds out of it. With mycoponics, we can just grow the mycelium on the tube, scrape it off, and it grows again. So we have a platform now for continuous bioproduction of about 18% of the world's current pharmaceuticals. But the other thing we can do with the mycelium growing on the tube is actually use it as a material system too. So those mycelia we can grow into a mycoleather. So we actually have a mycoleather platform. This is a three-dimensional mycoponics tube, and on top of this we can grow a mycelial leather material that then we could peel off and use as a glove. So we're actually looking at growing personal protection equipment, and if we can impart the radio resistance that's innately part of many fungi into the mycelium, we can actually have radiation shielding materials that we can grow in space. And again, we can grow those from waste materials. We're planning on growing that from urine.
Cool. So, like, if you can gene edit the fungi to and include a D-sub gene inside, which is for the tardigrade, you can also make it like radioactive protected.
Yeah, many fungi are already radioactive. I mean, radio resistant.
Resistant, yeah.
Yeah. So there were some fungi they also recovered from Chernobyl. A radiation resistant mycelium that seems to be growing from the radiation in Chernobyl. But that's not uncommon. That's really pretty common that most fungi have this radio resistance, and that's not well understood, although I think it has to do with their mitochondrial systems. And that's one of the things I'm interested in studying now, based on using mycoponics and what we've been able to do with the mycelial system, growing it on the mycoponics. we actually have been doing some really interesting new biothermodynamics with the mycelial system that we think relates to their mitochondrial systems.
Okay, cool. Marshall, let's get back to your most recent policy paper. The paper makes the direct assertion that China has now surpassed the US in the scale and the preeminence of these BLiSS bioregenerative technologies. Given that China's lunar program is advancing rapidly and they are actively recruiting international partners, does the US risk being left behind on the race of building sustainable habitats on the moon? And what are the geopolitical implications of the next great leap in sustainable living if it's demonstrated by not by NASA, but actually by the Chinese National Space Administration?
If you go back and look at the origins and roots of what was the Soviet space program when it was born, it actually all came from a Russian philosopher and technologist by the name of Konstantin Tsiolkovsky, who lived between the 1870s and early 1920s. But essentially, if you go and take aerospace engineering and study at a Russian university today, you will learn Tsiolkovsky's mechanics. So
Okay.
when they flew Sputnik, they did it all based on Tsiolkovsky's mechanics and his background. He was also a visionary who envisioned bioregenerative life support and wrote books on this in the 1800s. And so when the Russian program started, the Soviet program started in the early 1950s, they had already started a whole program in bioregenerative life support in Siberia, and also another program at the Institute for Medical Problems in Moscow. But they've had ongoing programs in bioregenerative life support and had multiple missions and trials with closed systems with people inside for a number of years. And the full extent of that is not known. Now, when we were working with the Russians and with the International Space Station, we benefited at the time because we didn't have launch ability to get crew to the station. So we collaborated with the Russians, and that was really actually, I think, a really great for NASA and great for the American program, because we really did learn a lot about space stations by working with the Russians. Because when the Soviet moon program failed, they just pivoted to their space station program. They already had a long and ongoing space station program at the time that they continued on. So by the time, Mir was here, that was their fourth space station. So they have tremendous amount of experience, working experience, theoretical and practical experience, living and working long-term in space, which we benefited from in the space station era. But now they have shifted over to collaborate with the Chinese and the International Lunar Research Station. So they're official partners now of the ILRS, and bringing with them all of that expertise, which includes decades of closed environmental bioregenerative life support research that they've conducted out of their facilities in Siberia. So that's the Russians are really quite potent still in space, even though they've had to deal with very, very limited resources. What they've been able to accomplish with those limited resources has been outstanding, and they're going to take that experience and knowledge with them in the collaboration with the Chinese.
Okay. Like you've made in your paper and here on this podcast, you've made a really compelling argument about the importance of the bioregenerative life support system. Also, your paper highlights spin-off benefits of this research for Earth, from advanced agriculture to environmental resilience, aligning with the United Nations Sustainable Development Goals. How does developing a closed-loop system for Mars provide a direct blueprint for creating more sustainable closed-loop systems here on Earth?
So when we're working on and developing bioregenerative life support, and we're considering humans in these life support systems, we think about that as we would be thinking about any human on this planet. What do you need in terms of food, water, waste, clothing, toothpaste, medicine? How does your medicine end up in your waste? How does that affect the wastewater treatment? So that focus on closure of the system and closure of all the needs that the individual needs, but also all the waste materials and recycling that. That's really, we don't recycle our waste. We don't live in human civilization is not closed in terms of its impact on the global ecology. In a sense, we rely on it. We've been dumping into it our entire history of our civilization, especially with our fossil fuels, dumping into the atmosphere, shifting the redox. So the technologies we need in order to, close ourselves in terms of having an energy source that's, renewable, but then using that energy input to be able to renew and recycle all of our inputs and outputs. That's the very technology we need for sustainable civilization here on Earth. So literally, I believe that all the biology that we're going to need to integrate into a bioregenerative life support system is going to include a lot of factors that weren't imagined or understood in terms of all the biological connections between all the biological systems on Earth. We're going to have to figure out what all those little factors are and how all that exchange is happening across the whole system, so that we don't end up with one little tiny thing that goes off scale later on.
Okay.
That's how tuned we have to be. Mm-hmm. Now, if we get that tuned in space, we're going to be that tuned on Earth in terms of our sustainability and our
food waste, plastics,
our resiliency, our regenerative nature of our cities, of our homes, of our civilization within the planet. We should have the same type of position within the planet as we would have on the moon. That is almost closed, where we don't need anything from the outside, so we're not affecting anything on the outside. We really need to adopt those types of strategies in order to save the Earth, but we really have to embrace the ecological connections that make those types of bioregenerative capabilities possible on the Earth, and recreate those in closed systems so that we can actually move that off the Earth. We're actually going to have to take life with us. We're going to have to take the essence of life on the Earth with us, or we will never survive, because we are a biological product of this planet, and we will never be able to survive without the biology of the planet. A human being would not be able to survive if you could isolate a human being from all other microbes and have an aseptic human. That person would die. Some horrible, disgusting, painful death.
Yeah. We're more of a super organism rather than just an individual.
Yeah, we are an ecosystem, and our ecosystem of our body is a continuum of the ecosystem we live in, and we can't cut that.
Mm-hmm.
We're going to have to figure out how to recreate that ecosystem in a closed, regenerative way, or we're not going to be able to go. In order to figure that out, we're going to have to figure out how to save the planet at the same time.
Yeah. So in your paper, you're not just making an argument for the need for the BLiSS system. You also have a hope and a plan. So the 2023 Decal survey and your own paper lay out clear recommendations for the United States to regain leadership, including building a full-scale proto-flight habitat on Earth for testing. If you were given the authority and budget tomorrow to implement it, what would be the single most critical recommendation, and why is that the essential first step to getting the US back on track?
I think the number one goal is to establish bioregenerative life support capabilities, period. Whether it's on a one-person basis or a four-person lunar base that's going to be our next project, or conceptually even something bigger. I would favor picking a project that's actually part of a goal that's the next mission of the agency. So we should be going to the moon. We should have the goal of creating a sustainable habitat there where we can maintain continuous human presence on the moon with minimal resupply. And then we can go to Mars. Because right now we probably couldn't keep people alive during the trip. Just the radiation between
Mars,
Earth and Mars, we would have to build a shielded spacecraft, which we don't have a plan for yet.
Yeah, true. You also recommend convergent funding. Does this mean that the future of space life support is too big a challenge for NASA to solve alone, and it must become a national multi-agency priority like the Manhattan Project way back then?
I think really advanced life support, it does include almost every aspect of human life and human existence, including human health. So of course there has to be a human health component to this. We have human research program, and we have the goal of developing and advancing precision and personalized medicine. So that's one way that the larger mission incorporates the mission of NIH. But also, too, we need to have a complete agricultural system. And so we need controlled environment agriculture. And that's under the purview of the USDA. The USDA has funded a lot of the work in this area over the years, but so has NASA. And together, that did spin off a controlled environment agriculture industry, which is now a viable industry here and growing, although NASA never really took credit for that as a spinoff, in my opinion, even though they did the bulk of the work. But also too, if you look at humans and the human environment, it affects humans in space is going to involve science and technology that covers what every other agency does in some way, shape, or form. So it could be distributed amongst the agencies for human health, agriculture, environment, all the different various technology areas. That also includes in terms of human performance, health monitoring, environmental monitoring, sensing. It touches on every aspect of human existence and human life. So there's no reason why it shouldn't actually be distributed amongst all the science agencies, because literally all those science agencies were created because of Sputnik to begin with. We wouldn't have the NIH, the NSF, and the national science infrastructure that the U.S. has today if it wasn't for Sputnik. I mean, I actually teach this in my space policy course.
Okay, so we see talking about influence from other countries and collaborations and competitions. We see ESA's Melissa program, the DLR's collaboration with China, and the new global initiatives like the Space Food Consortium are emerging. In the new multipolar space environment, is the path to developing these critical technologies paved with fierce competition, or is a global collaborative effort and an ISS, an International Space Station model for life support, the only way to truly succeed?
A lot of this has been done as a global effort, and in the spirit of collaboration. There's been large numbers of shared projects and collaborative activities between NASA and the European Space Agency and the Canadian Space Agency. They also have programs in this area and research that they've funded in these areas in the past. So I really see that's really part of the U.S. and global national effort, really kind of an extension of the ISS research community into what would be, essentially an international lunar outpost. So potentially led or um, um, as a foundation partner would be the United States. And so I see that as being international, and I see that there's still a lot of interest from other national agencies in controlled environment agriculture outside of space. So a lot of that work gets done in the context of agricultural and biotechnology. So there are a lot of um, funding agencies that fund that type of work, but for terrestrial applications. But they could be funding it in the context of terrestrial applications, and there could be more than one pool supporting this type of research so it can move forward faster.
Okay, so much of the groundbreaking work that we talked about today happens at the physiological sensing facility at Purdue, which you co-direct. For the students and researchers listening, what is the role of an interdisciplinary hub like the PSF, and how does bringing engineers, biologists, and nanotechnologists together under one roof accelerate the pace of discovery compared to traditional siloed academic departments?
Yeah, so we work, broadly across different types of material systems and also across different engineering disciplines to some extent. And then within our application area of life sciences, we work in microbial, fungal, plant, human systems. So USDA, EPA, NIH. So we end up with, different collaborators and different partners and different students coming from different backgrounds and training coming in and collaborating together. Sometimes it's a horticulture student working on some plant science project with a sensor and working with an electrical engineer. And electrical engineers learning about the plants, and the plant biologist is learning about the sensing systems and how that works. So that's one way that you get cross-pollination across disciplines. But a lot of times I end up bringing in a student that has a master's degree or undergraduate degree in electrical or mechanical engineering, and they're going to do a biology project. Well, I force them to learn the biologist. And by the time they finish their degree and more advanced degree in our lab and in the context of agricultural and biological engineering, they are engineers and biologists at the same time. So I believe in interdisciplinary work. But I don't think it should be sugar-coated. I think you have to get in and actually do the hard work to cross the disciplines. If you're dealing with an electrical engineer or another scientist or a colleague from another discipline who doesn't understand your language, and sometimes the same terms have different meanings in two different fields. And I've seen people get in these fights when it was just really a definition problem. So, but it's really rich in terms of what you get in terms of outcomes, because when you start mixing engineering and science and physics and chemistry and biology in unique ways, you always learn.
Great. Marshall, looking over the next 15, 20 years, as we prepare to send humans back to the moon with Artemis and then onwards to Mars, and as you look towards the future, what is the one fundamental question about how life works in space that you are most personally driven to answer before you hang up your lab coat?
The number one problem we're focused on in the Artemis mission will be the radiation environment, specifically looking at how radiation interacts with energetic organelles. So in my previous work with Chromex, I was working with the plant root systems, and we were seeing these biophysical responses that were associated with the biophysical environment, but could be radiation. We weren't able to actually discriminate that in that work, but in the leaf mission, that's going to be our primary focus, because the plants are going to be growing they're going to be in a box, so they're not going to be right out on the moon. So they're not going to be directly getting radiation. They're not going to get any solar radiation. We're going to have lighting systems, but that box is going to be transected by the lunar radiation environment. So it's going to be high fluency environment, and how that radiation is interacting with the chloroplast and the photosynthetic apparatus of the plants is really going to be really my main concern and focus. I believe that those radiation effects that are mediated through through organelles are the primary mode for stress and pathology in space, whether it's a human, microbe, fungi, or a plant. It is mediated through these organelle systems because the organelles amplify the damage. If you have a particle come in and smash into a phospholipid and knock a hole in the membrane, well, the membrane just closes back up.
Okay.
But if it smashes into a mitochondria and creates a reactive oxygen species event that actually kills that cell and then the three neighboring cells around it, that's different.
Okay.
And that's the kind of damage that you can get through these types of radiation events when you start interacting with these energetic organelles, because when they're damaged, they can actually continually spew out reactive oxygen species. So it can be it can amplify the damage. So it's really focusing on those sources of damage because those are the most profound in terms of long-term effects.
Cool. So before we wrap up, is there anything else you'd like to share with our audience or advice you would like to share?
I think that the work that we do, focus on bioregenerative systems, is really important because of how that will solve the major problems for space exploration and enable humans to actually go and actually inhabit and explore these off-world extraterrestrial locations. I believe humans have a place off this planet. I believe that the future of human civilization involves space exploration, and humans are going to move out into our solar system and hopefully beyond at some point. And it's going to require this type of knowledge, this type of mastery of biology. Can you master the biology of the planet you were born on? Or can you master the biology of the planet that made you? Because if you can't preserve what made you, you will not exist in the future. We're going to have to take a subsect of the planet with this when we go to explore outer space. And that's really what it takes. That's really what it takes to be a starfaring species. You have to embrace and love your home first.
Yeah, that's a great way to finish this episode. Marshall, it's been an honor to have you here. Thank you so much for sharing your insights and expertise.
Thank you so much, and I'm sorry for all the confusion we had in between, but
no, don't worry about it. To everyone else listening, this is the Omar Aleli podcast. Thanks for joining us and keep exploring.